All right, welcome back, everybody. Coming at you today with the last part of Chapter 7. All right, and so we're going to pick up where we left off. The first thing I want to talk about is something called zymogens. Zymogens are defined as inactive precursors of active enzymes. Enzymes are not always just sitting there in the same exact form, just ready and waiting for a reaction to catalyze. They actually have to go through different processes, right? A lot of enzymes are sitting around in an inactive form that's actually in which they've actually been rendered inactive, not through one of the previous means we've mentioned, but because they're actually in a covalently bonded form that just doesn't work, right? So, another way of activating certain enzymes is to actually cause one or more covalent reactions within the enzymatic chain, right? Think of it a lot like a bush, right? A hedge or something, right? You can have your hedge in a certain form, but you can also trim it. And there are people who make these wonderful little topiary structures that have animals and shapes and all sorts of things, right? That's kind of what zymogens are. Zymogens are the bush, and they can be trimmed in certain ways to make them into a new form that is actually biologically active. And we're not going to sit here and look at every single zymogen. There's just way, way, way too many of these to look at. Look at them all, but we're going to go over a few examples. And the one that we're really going to go over most in depth is something we've already mentioned, and that is chymotrypsin. All right, so previously we talked about chymotrypsin. Can you remember the context? Can you remember what chymotrypsin was used for? Give you a second to think about it, or perhaps look back in your notes and find out. Hopefully you paused the video and looked that up or thought of it. Chymotrypsin is used to cleave peptide bonds in proteins. It's used to help digest proteins. Specifically, it likes to cleave peptide bonds that have amino acids with aromatic side chains, such as, for example, phenylalanine or tyrosine or tryptophan, right? Well, chymotrypsin is not always active. It only is activated when it needs to be. All right, so let's take a look at this. All right, first of all, let's take a look at the inactive form. The inactive form is actually not called chymotrypsin. It is called chymotrypsinogen, and does have not only a different tertiary structure, but also a different secondary structure and primary structure, right? We're actually going to be sort of pruning the amino acid chain here to make it a little bit different. All right, so chymotrypsinogen can itself be cleaved, right? We know it cleaves other proteins, but it itself can be cleaved. And it is cleaved by an enzyme called trypsin. So I'm just going to, one second. I'm just going to start writing some of this down. So we have chymotrypsinogen. It gets converted into chymotrypsin. Chymotrypsinogen is inactive. And this one is active. And this reaction is catalyzed by an enzyme called trypsin. Okay, so what does trypsin do? Well, I'm going to preface by saying, actually, this isn't the final form of chymotrypsin. This is actually an intermediate form called pi-chymotrypsin. We'll come back to this in just a minute. All right, so what does trypsin do? It is going to cleave the chymotrypsinogen chain at arginine 15. All right, and this is what is called pi-chymotrypsin. All right, so if you look a little bit further on, and again, you'll want the complete picture on a real computer or your book. Here is a representation of both chymotrypsinogen and pi-chymotrypsin. All right, so you have the peptide chain here, the amino acid chain. The little yellow lines, those represent disulfide bridges. Remember those? Yeah, disulfide bridges. They're formed between thiol groups, and that's usually going to be between a couple of cysteine residues, right? So you have a couple of disulfide bridges, and you also have your sequence. And if you look at 13, 14, and 15, after 15, that's where we're going to get that initial cleavage. That's an arginine residue, right? So trypsin is going to snip chymotrypsinogen at arginine 15. These are still attached, right? These are still attached by the disulfide bridges, but there is sort of a gap between 15 and 16 here. This is pi-chymotrypsin. And it is an active enzyme. However, it's not active on other proteins yet. Pi-chymotrypsin actually is active on itself. It catalyzes its own transformation into the next stage. So it is self-catalyzed. And this gets converted into alpha chymotrypsin. And this is the final active form of chymotrypsin. That's the one that actually does the job that I told you about previously, where it likes to help digest other proteins at areas with aromatic amino acid residues. Okay? So, again, if you look back at this picture and you see the full picture, hopefully in your book or PowerPoint, basically what happens here is pi-chymotrypsin will self-remove 4 amino acid residues, right? It's going to snip out serine 14 and arginine 15. And then it's also going to snip out threonine 147 and asparagine 148. All right? And that basically renders the entire chymotrypsin chain into 3 segments, 3 pieces, right? However, they're still all connected to each other through disulfide bridges. This specific arrangement will allow the polypeptide chain to arrange in a conformation that makes the active site that can fit incoming proteins that it has action on, right? So it can't do this until, it can't actually do its job until it gets into this form, right? Well, what signals this? Everything is a signal in biochemistry, right? There's so many start and stop signals from one pathway to the other and everything. So basically what happens is when you eat, right, we consume a protein and it reaches the small intestine, that basically signals the activation of trypsin. And then trypsin will start to do its thing and cleave chymotrypsinogen and form chymotrypsin. But that's not triggered until you actually get the protein coming into the small intestine, ready to be digested. So, it's pretty cool. Why does this particular active site work, or the others don't? Well, here's a little bit of chemistry for you. Favorite thing, woo, chemistry. All right, so when you snip out that those residues 14 and 15, that basically exposes residue 16 as the N-terminus of this second chunk, the second free peptide chain that's being held together with disulfide bridges, right? This new N-terminus of this part is isoleucine. All right, and in this environment, that isoleucine, its N-terminus is going to be protonated. So you're going to have an NH3. I'm just going to draw it like this for simplicity's sake. All right? So you have a protonated NH3 on your isoleucine. You know, the reason I keep making these big stripe marks here is because I try to swipe in order to get rid of this and get it to the other stuff, get back to the PowerPoint, but for some reason it never registers, so that's why it keeps happening, in case you're ever wondering. All right. Okay, so we have that protonated isoleucine. Now, long way down the line, we have an aspartic acid 194. All right? Way down the line, but due to the folding of the entire chain, aspartic acid 194 comes into close proximity with isoleucine 16. And remember, aspartic acid has that side chain that is an acid group, right? It can exist in a deprotonated form, right? So when aspartic acid is deprotonated, we basically call it aspartate. It refers to the same thing, it just means that its side chain is deprotonated. So we have that aspartate 194, and it's got a carboxylate residue hanging off. All right, so remember all those different forces we talked about that can help hold together a protein's tertiary structure? One of them was hydrostatic interactions. That's just another fancy schmancy term for ionic interactions. Plus charges being attracted to negative charges, right? So these are going to form an attraction. It basically forms a salt, sort of like Na plus and Cl minus, right? So this forms sort of a salt bridge, and basically this salt bridge between these two amino acids is key. This is key to formation of the proper shape conformation of that active site. If you don't have this ionic interaction, you can't make the right active site, okay? If it's not in alpha chymotrypsin form, you don't have the ability of that protonated isoleucine to come into contact with your negative aspartate 94. So it's really neat and kind of mind-boggling to think about it, but it's very much a domino effect. Things got to happen in a certain order in order to get the enzyme in its correct activated state. Okay. Like I said, we're not going to go over every single zymogen, and that would take forever. But just to highlight a couple of other ones, we mentioned trypsin, right? Trypsin itself is not always in an active form. It is originally in a deactivated form called trypsinogen. It's activated into its active form of trypsin by an enzyme called enteropeptidase. Much in the same way trypsin acted on chymotrypsinogen, right? It kind of snips certain portions in order to get it to eventually get into the right active site conformation. Couple of really interesting ones. Thrombin, for some reason I always have trouble saying that word. Thrombin and fibrin. These are both very important to blood clotting. All right. I'm going to show you a picture. This is not in your book. I just Googled this. And I'm not expecting you to know this. This is something like if I were to teach a biochemistry too, this is something I would typically have you learn. But blood clotting is a fascinating process. It's very complicated. You cut yourself, you start to bleed, and after a few minutes, unless it's just a really, really, really bad cut, it'll stop bleeding, right? It'll clot. Stick a Band-Aid on it, put some pressure on it, it'll stop. That's because the blood will clot. You'll have a bunch of these red blood cells that'll sort of stick together to literally make sort of an artificial plug for the wound, right? Now, the processes that go into this, the individual processes that go into blood clotting, is very complex. This is a diagram of something called the blood clotting cascade. And these are really all the different steps that have to happen to get your blood to clot. And you see a bunch of these are labeled factors, factor 12, factor 11, factor 9, factor 7A, etc., etc. These are known as blood clotting factors. Okay, I'm not going to get into the weeds here. Again, I want to show you this just in context of both thrombin and fibrin. If you look down below here, really close to the end, right? You see the second from the bottom, you have factor 2. Factor 2 is prothrombin, the zymogen prothrombin. It is activated by a couple of other factors to transform into factor 2A, which is thrombin. Factor 2A, thrombin, can then act upon fibrinogen, which is a zymogen form of fibrin. And then once you form fibrin, then the fibrin can clot, and you can actually heal your wound, or stop it from bleeding at least. So, pretty neat little, it's really kind of awe-inspiring. I'm kind of geeking out a little bit here, but, I'm a chemist, so I'm allowed. It's really neat. All right? So, all sorts of metabolic pathways have these zymogens that have to be activated by some other species / some other event. Right? And these are just a few examples. All right? But right now, I want to dive a little bit deeper again into chymotrypsin. So we said it has to be in a certain orientation. You got to form that active site just right in order to bind the incoming species. Why is that? Why do we have to get it in a certain way? Well, to understand that, we're going to use chymotrypsin sort of as a model, and we're going to look at the actual chemical reaction. Brace yourself. I'm getting giddy here because guess what we're about to do? We're about to do organic chemistry mechanisms. It does my heart good, and you guys are probably just groaning right now. Both in terms of dreading this and also cringing at my geekiness. So I have no regrets, and I'm going to show you how this works. All right. So, chymotrypsin. First of all, when you're trying to figure out what's happening with an active site, you got to know which residues are the really important ones, right? Whenever you make an active site, of course, there's going to be a little pocket of amino acid residues. Usually, there's just certain ones, though, that are really key to actively making the reaction go, right? How do you figure out which ones? Well, it's a painstaking process. Basically, you got the polypeptide chain, and you have to go through a process called labeling. This is where you take each amino acid and sort of block off its activity, right? You sort of react its side chains with some sort of agent that means those side chains are not available to react when a substrate is present. Sorry, I had the hiccups. So, yeah, that takes a long time. So, somebody took chymotrypsin and went through the process of labeling each one of its amino acids by blocking it off and then seeing if it still did the job it was supposed to do. So, basically, after all this, they found that histidine 57 and serine 195 were the critical amino acid residues. I'm not going to go too much into how they labeled them. They are here. Serine 195 was blocked off by something called diisopropyl phosphofluoridate, or DIPF for short. And basically what that did was it reacted the OH on the serine with a phosphate group and made a phosphate ester, right? Histidine 57 was blocked off with an ungodly sounding name, N-tosylamino L-phenylethochloromethylketone. Rolls right off the tongue, called TPCK for short. Well, histidine, of course, its characteristic feature is this 5-membered ring with a couple of nitrogens in it. And so basically, you formed an inactive amide with this compound, meant it couldn't have action. So again, I'm not really that worried about you guys knowing these exactly, but it's kind of interesting to see how they were labeled. But yeah, well, you do need to know that these are important here. So we're going to see what actually happens at that active site. Remember, what does chymotrypsin do? It cleaves peptide bonds. So we're going to show how it cleaves a peptide bond of an incoming peptide or protein. All right. So here's the mechanism of chymotrypsin. Now, this picture is way cut off on the iPad. You should be able to see it on the computer and the book. All this is written out for you, but I'm actually going to draw it for you on the whiteboard as well, because, you know, this is a philosophy I have with organic chemistry and mechanisms. Even if I have a picture of it in a book or on a slide or something, there's a lot of muscle memory involved. This is why I always really harp in OChem on drawing mechanisms, because you really learn how to do it there. So I'm going to show you by hand here, and hopefully you will follow along, hint, hint, because that'll help you remember it. All right, so let's take a look. So it starts off with serine 195. That's one of our important enzymes. And you know, it's got an OH hanging off as part of its side chain. I just realized I need to draw it more like that. All right, and then you have a histidine, histidine 57. And again, I'm only going to draw part of it. You have part of it is this 5-membered ring present in the side chain. Okay. And then lurking off in the distance, I'm going to go ahead and represent that incoming peptide. Okay, so this starts off because, by the way, that should be 57, not 55. All right, so the way these are arranged, this histidine is in pretty close contact with, close proximity with your serine 195. Because of that, this nitrogen on this ring, it can start exerting a little bit of a pull on that hydrogen. It can start tugging it away from oxygen a little bit, and that's going to make this oxygen a little bit more nucleophilic. This is essentially just a nucleophilic substitution that occurs, right? If you're tugging this away, that's going to expose these electrons a little bit more, make this more negative. And what's going to happen is this is going to attack the carbonyl carbon in your peptide bond. And that's going to cause, temporarily, a pair of electrons to hop up here. All right? And then once that is said and done, you're going to have this. All right, sorry it took so long for me to draw that, but it took a little bit. Okay, so now at this point we've gotten the oxygen on the serine directly attached to the carbonyl carbon of your peptide. The H is now more associated with the nitrogen in the histidine ring, and then loosely associated with the oxygen of serine. So you're going to see sort of a tug of war that happens for that hydrogen. And you have this negative carbonyl oxygen. Okay? Well, remember I said that pi bond move up to the carbonyl oxygen was only temporary. It can actually just fall right back down and reform that double bond. And when it does so, it's going to kick off this nitrogen group. Right? That amino group is your leaving group in this case. Right? And so this nitrogen is going to have an extra pair of electrons as a result. And as a result, it's going to jump up here and it's going to grab that proton. Bear with me here. I'm going to erase this part so I can continue. All right, so what does that leave you? You have the serine residue, still got that oxygen attached, and that oxygen is still attached to your carbonyl carbon. That carbonyl carbon has a double bond, and it's still got that R1 group, right? So this is a ketone. No, excuse me. Not a ketone, that's an ester. Sorry, my bad. So, at the end of that step, serine 185 has formed an ester with the carbonyl group of your peptide. All right? Your histidine is basically just sitting there like it always does. It's really ugly, by the way. Sorry for that ugly double bond there. All right, it's just sitting there like, in its normal structure, unaffected. And that amine group that was originally part of your peptide has now been kicked off as its own product. Right? So basically, this is the halfway point of the reaction. At the end of the first halfway point, we've basically snipped off the amino group from your peptide and set it free. Right? So now we start the second stage of reaction. Here's a little hint. Remember, a little thing about catalysts is that catalysts are always regenerated. Catalysts always end up in the same state as when you started. Enzymes are no different, right? Enzymes are natural biological catalysts. So the second stage of reaction here is going to be in large part getting them back to their original state. All right, so let's see how we do this. Again, let me kind of get rid of some of this to get some more room. All right, so as we begin the general second stage here, something else comes along. A little something I like to call H2O. That's right, it's water. Water is the primary reactant in the second part of this process. Okay? So basically what happens here is nitrogen. Again, nitrogen's an electronegative atom. You got a couple of electrons sitting on nitrogen. It's going to see that partial positive on hydrogen of water and say, hey, I'm interested in that. So it's going to sort of exert a pull on that hydrogen. That's going to make this oxygen even more electronegative than it already was. And as a result, it's going to attack that carbonyl carbon and promote the electrons, again, temporarily, All right, so when it does that, you get this. Okay. All right, so this oxygen, this water is attached to your carbonyl carbon, right? The H, remember that H that was sort of attracted here to this nitrogen? Well, at this point, it's almost completely gone over to nitrogen, so much that the nitrogen gets a positive charge, right? Still loosely associated with the oxygen here. We're going to finish up by one more little arrangement, all right? So what's going to happen is this pair of electrons is going to come back down and reform your carbonyl. That's going to give that carbon, the carbonyl carbon, too many bonds. So what's going to happen is this bond is actually going to break. The one between that carbon and the serine oxygen. That's going to break. That's going to also allow that oxygen to just reach in and grab this proton. Okay? And when that happens, guess what? You've reformed your serine 195. That means this is ultimately released from everything. It's not connected to that H anymore, and it's not connected to the serine oxygen. So, it's free. Live free. Woohoo. And it's released as a carboxylic acid, which will probably get deprotonated just in the pH environment that it's in once it's released, and it'll eventually form carboxylate, right? But the acid, the full acid, is the leaving group. And then your histidine no longer has any hold on that hydrogen anymore, so it goes back to its original form. And we've reformed our original amino acid residues, and a series will start anew with another peptide coming in. So, good example of what typically happens in active sites. They're not all nucleophilic substitutions, but a lot of them are, right? So that's fairly representative of a lot of the things that you'll be seeing. All right, so again, there's just boo-koodles of different types of enzymatic reactions that occur. I'm going to just highlight some of the ones here that we find. I'm not going to, go into all of these. We'll see examples of these as we get into specific metabolic pathways and metabolic reactions, right? Some of them involve acid catalysis, right? That's where an acid will donate a proton to kind of start things off. Some of them will have base catalysis, right? That's where you might have a deprotonation to start things off. You might have a Lewis acid catalyst. What's the definition of a Lewis acid? Let you think about that one for a second. Why don't you go ahead and pause this and look it up if you can't remember. If you can't remember, or if you've already gone and looked it up, I'll just go ahead and say it. A Lewis acid is an electron pair acceptor. And if you have something that can accept an electron pair to sort of kick off a reaction, that can be a Lewis acid catalyst. A really good example of this is carboxypeptidase A. This is going to be an enzyme that specifically cleaves C-terminus amino acids off. So you have an amino acid chain, right, a peptide chain, and it'll just position itself over on the C-terminus end, and it'll just start snipping them off one by one from that end, right? That's what it does. Carboxypeptidase acts via a Lewis acid catalyst mechanism, and that's because it's got a metal ion. Again, you can't really see it here, but if you look in your PowerPoint on the computer, you can see that carboxypeptidase, part of its structure is actually a zinc, right? Zinc 2, all right? Zinc typically has 3 bonds here, as you can see, in its normal state. It's holding 6 electrons. Guess what? It can hold more than 6 electrons. So what happens is, as a peptide comes in, it will seek out the carbonyl group, and it'll interact with the carbonyl oxygen, right? Remember, that oxygen's got a couple of electron pairs, so one of those pairs can be donated to zinc, and it can sort of make this complex. And that helps open things up to start the rest of the reaction. That makes the carbonyl carbon more susceptible to attack by water, and the rest is history. All right. Speaking of metals, we can also see metals pop up and be helpful even if they're not actually part of the enzyme structure, right? They're very, very helpful. We'll see. They're important in the blood clotting cascade that I mentioned, and they're important in a lot of other things. Metal ions can be used in this manner as what we call a cofactor. A cofactor is going to be defined as a non-protein substance that sort of helps out. It helps out enzymes when they catalyze reactions, right? It's their little helpers. And cofactors, just like enzymes, are going to be regenerated in their original form at the end of the reaction, right? Metals are really prominent in this. Also, organic compounds called coenzymes are pretty plentiful in these roles. A cofactor is just simply an organic compound that helps out enzymes, but they're not actually proteins themselves. They're going to be some other type of organic compound. Vitamins, right? A lot of coenzymes will come from vitamins. Not all of them, certainly, but a lot of them. Especially vitamin B. It tends to be a really, really good source. And you can see an example of just some of the more prominent coenzymes in this table, and you see they have a wide array of functions, right? And some of these you've probably heard of, either from your biology classes or just simply as kind of being nutritional, products or buzzwords or something that you've heard in marketing. Biotin, right? I'm sure you've probably heard of biotin. It is actually used to help in carboxylation reactions, adding carboxyl groups to particular compound structures, right? Coenzyme A, you've probably heard of that before, right? If you haven't, trust me, you will. That's used to transfer acyl groups or carbonyl groups from structure to structure. Lipoic acid, again, also used for transfer. Flavin coenzymes used for redox processes, et cetera, et cetera. I'm not going to go through all of them. I do want to highlight one in particular to sort of use as an example because it's one that you've probably seen before or heard of. That is nicotinamide adenine coenzymes. They are derived from the vitamin niacin, and they're involved in redox reactions. You may ask yourself, what the heck is that? That does not sound familiar. That may not sound familiar, but a little something called NAD plus might sound familiar. NAD plus and also maybe NADH, right? You've probably seen that in several of your biology classes, right? So this shows the structure of NAD plus. Again, look at the full picture. It's actually got actually I guess four major parts to it, right? At the top of the structure, you see that fused ring system. That is adenine, and yes, that's the same adenine that you find in adenine triphosphate. Yeah, ATP. You have a couple of ribose units. Those are these five-membered rings with oxygen at the top. There's two of them. Ribose is a simple sugar, and we'll talk more about those when we get to chapter 16. You have phosphate groups linking the two ribose units, right? So you see a phosphoric acid anhydride linkage. And lastly, you see the nicotinamide group. Nicotinamide is going to be the six-membered ring. It's aromatic, and it contains nitrogen as one of its members. And it's also got a primary amide group on the side, carbonyl with an NH2, right? That's where the term amide and nicotinamide comes in. And this is where the plus in NAD plus comes from, right? Because you see that nitrogen in that nicotinamide ring, it's got 4 bonds, so it's going to have a positive charge. All right. NAD plus can be reduced, right? What happens when something gets reduced? It gets more negative, right? So we can actually see a conversion of NAD plus into NADH, which I'm sure is something that you've heard of before when discussing metabolic reactions in biology classes. Basically what happens in the reduction of NAD plus is that essentially, for all intents and purposes, I'm going to simplify this for you a little bit. For all intents and purposes, a hydride is added to the structure. And that's H minus. That's H with a free pair of electrons and a negative charge. You can see it in this middle structure. Technically speaking, that's not how the mechanism works, but again, we're not worried about the mechanism here. I'm just sort of showing you the net effect, right? So basically what happens is that H minus gets added to one of the carbons in the ring, and it causes a series of electron movements, some resonance to occur. And as a result, a pair of electrons gets donated from that carbon-nitrogen double bond in NAD plus to that nitrogen by itself. So now that nitrogen only has 3 bonds instead of 4. All right? So that's why we call it NADH, and that's why it doesn't have a charge anymore. We've essentially added 2 electrons to the system. If we get to it, and I don't know if we will or not, we might see this in more detail later on. Depends on how far we're able to get in the class. All right. Just showing you, some of the other B6 vitamins. I mentioned these before. Here are some of them. I'm not concerned about you knowing the structures of these or anything. You should be familiar with the names and what they are, right? You see a lot of commonality here in the names. Pyridoxal, pyridoxamine, pyridoxine. All these have pyridox in them. They're all kind of derived from the same original structure. And they're all actually involved in transfer of amino groups. They can pick up an amino group from one compound and stick it on another one. So, this last slide sort of shows, and one of the several examples of this, right? You can actually have a couple of compounds here, glutamate and pyruvate, and you see glutamate has an NH2 kind of sticking up out of it. Well, transaminase, with help from one of these coenzymes, pyridoxal phosphate, can basically pick that NH2, stick it on this pyruvate over here, and turn it into an alanine, and a carbonyl group ends up getting transferred to glutamate to turn it into alpha-ketoglutarate. So, just an example. of how those occur and basically what its function is. You'll see as we go along, enzymes, classes of enzymes, often have very particular functions like moving an amino group, moving a carbonyl, whatever. But coenzymes, in their helper roles, also have very defined things that they do. All right, that is it for Chapter 7. I enjoyed this. This is because it had a lot of organic chemistry, so I'm sure you enjoyed it too. All right. Look forward to talking over this. Any questions that you might have. If you have any questions, let me know. All right, until next time, have a good one. Bye-bye.